This is Mrs. Berkeley, and I'm going to review some information that you will need for your biology end of course exam in Florida. This video will review DNA as a common genetic code, protein synthesis, proteins, and mutations in DNA. Please watch the video for DNA replication before viewing this video. DNA provides the instructions for all organisms to grow, survive, and reproduce. DNA must remain in the nucleus of a eukaryotic cell. Recall that eukaryotic organisms include plants, animals, fungi, and protists. The nucleus protects DNA from damage that might happen from all the activity taking place in the cytoplasm with other organelles. DNA instructions must be converted into messages for the organism to function. This process is called protein synthesis. Synthesis means that elements are combined to produce a new product. Think of it this way. If you worked in a shoe factory, you would not make shoes, you would synthesize them from cloth, rubber, metal, plastic, etc. For proteins, amino acids are linked together to produce a protein. Protein synthesis begins with DNA unzipping the required section. The instructions in the DNA code must be converted to RNA. RNA also has nucleotides composed of a phosphate group, a 5-carbon sugar, only this time it is called ribose instead of deoxyribose like the sugar in DNA. There are 4 different possibilities for nitrogenous bases in RNA: adenine, uracil, cytosine, and guanine. Notice that there is no T in RNA. C and G are still complementary to each other, and T will match up with A, but A will now pair with U. Transcription takes place inside the nucleus of a eukaryotic cell. Once the DNA is opened up, complementary RNA nucleotides come in to form a polymer called messenger RNA or mRNA. Let's try a coding exercise. Remember that this time we are using U instead of T because this is RNA. Okay, once more. mRNA is read in groups of 3 by ribosomes. The group of 3 nucleotides, or triplet, is called a codon. Scientists have figured out which amino acids attach to each codon and developed this chart. Let's say that you are trying to find the amino acid that matches up with the codon AUG. A is the first nucleotide in the triplet, so it is the first base. Locate the A, which means that our amino acid will be somewhere in this section. Now locate the second base. Since U is our second letter in the triplet, we have narrowed down the search for our amino acid to this section. Now for the third base in our codon. The letter G means that we know our amino acid is MET. Messenger RNA exits the nucleus and goes into the cytoplasm, where it meets up with a ribosome that clamps onto the messenger RNA. tRNAs bring in an amino acid, match up with the appropriate codon on the mRNA, and drop off their amino acid. Remember that translation takes place in the cytoplasm. Amino acids will link together to form a polymer called a polypeptide. Polypeptides fold together to form proteins. To summarize, protein synthesis takes place in two stages. The first stage is transcription, which takes place in the nucleus. In this step, DNA is copied by messenger RNA, and then mRNA exits the nucleus and moves into the cytoplasm. The second stage is translation. It takes place in the cytoplasm. mRNA is read by the ribosome factories, where tRNAs bring in amino acids to form polypeptide chains, and later polypeptide chains will fold together to become proteins. You will probably remember the order of the processes, but the words might be difficult to remember. The easiest way that I have found which term comes first is that C comes before L in the alphabet, and transcription comes before translation. Proteins are responsible for most of the biological processes in our body, including tissue growth, movement, the immune system, and biological functions. Most enzymes are proteins. Wait, let's review our macromolecules again. Macromolecules are large molecules. The prefix macro means large. Biological macromolecules fall into four categories: proteins, carbohydrates, nucleic acids, and lipids. Many biological macromolecules are polymers. A polymer is composed of many monomers. If you think of a monomer as one of these beads, the polymer is the chain of beads. The prefix poly means many, and the root word mer means part, so a polymer is many parts. Since mono means one and mer is still a part, monomer means one part. So proteins are macromolecules. They are polymers. They are composed of many amino acids. Shape or structure affects function. You could choose from many different shapes to place underneath a car, but the one that works best is the round one if you want it to roll easily. If you change the shape, it will no longer function as well, if at all. If the shape of a protein is changed so that it no longer functions, it is said to be denatured. Proteins can be denatured by a change in pH or an increase in temperature. A decrease in temperature will slow down the rate of reaction. Remember that enzymes are proteins. You might see a graph like this. Every enzyme works best at a particular pH and temperature. If the pH is changed, the protein will denature. Enzyme A functions best at about pH 2.3. You see that function decreases as the pH changes. Enzyme B functions best at a different pH. Temperature also affects proteins. Enzyme C functions best at about 15 degrees Celsius, while enzyme D functions best at about 37 degrees Celsius. Perhaps enzyme C is in a bacterium that likes it slightly chilly, while enzyme D is in a human body. The molecules that an enzyme works with are called substrates. Every enzyme works on a specific substrate. That means that an enzyme will work on one and only one molecule. The enzyme names often end in ASE. Sucrase is the enzyme that works on sucrose. Lipase works on lipids. Penicillinase works on penicillin, and amylase works on amylose. An enzyme will act on a substrate to break it apart. The substrate enters the active site of the enzyme, and the enzyme acts on the substrate, allowing the bonds to break that are holding the two pieces together, releasing the products. The enzyme is ready to work on another substrate. Other enzymes will put two substrates together, making bonds that will produce one product. The concentration of enzymes and substrates makes a difference in the speed of the overall reaction. When enzymes outnumber substrates, increasing substrates will increase activity since the enzymes will have an easier time locating reactants to work on. When substrates outnumber enzymes, the enzymes have no difficulty locating substrates, but the reaction will proceed more quickly if you add enzymes. When enzymes and substrates are equal, adding more substrates will not speed up the reaction since the enzymes are already working as quickly as they can. Adding more substrates will allow the enzymes to work longer. Enzymes lower the activation energy necessary for the reaction to proceed. Think of activation energy like a push. The car needs a push to get up over the hill. Enzymes are like my hands lowering the hill so that it is easier for the car to proceed. Notice that the bottom of the hill did not change positions. Only the top of the hill was lowered so that it took less energy for my car to get over the hill. In a chemical reaction, the beginning and end of the reaction do not change. Only the amount of energy that it takes for the reaction to proceed changes. Enzymes can be used over and over again, just like my hands can be used over and over to lower many hills for my car. Sometimes the DNA is not copied correctly and a mutation occurs. Remember that DNA is used as a code to produce proteins. So if the DNA is changed, then there is a possibility that the amino acid sequence would be changed and the resulting protein would not function correctly. I am going to use a simple sentence to model some simple mutations. My original sentence is, the cat ate the rat. If a deletion is made, the resulting sentence does not make sense at all. The same type of mess happens if there is an additional letter or insertion that is placed in the sentence. All the downstream words are affected by the addition or deletion. This is called a frame shift mutation, and the resulting protein will not function. Sometimes a substitution is made and no other nucleotides are affected. The result is a slightly different meaning, but it still might function. The car ate the rat? Just a little weird. Now let's look at what you might see in a question. Notice that if you change a nucleotide in DNA that ends up as the third letter in the mRNA codon, the amino acid may not always change. There are amino acids that will use several different codons. The original codon was AAG, but the new codon AAA will still use the same amino acid, so it is a silent mutation, meaning that it will never be noticed because the protein will be exactly the same. Other times the substitution will change the amino acid. If it codes for a stop, the protein will not function. If it codes for a different amino acid, the protein may function differently than the original protein. Now for a quick summary of what we went over in this video. DNA is used as the instructions for all organisms to complete their functions. DNA is in the nucleus of eukaryotic cells. In order to carry out the instructions, the code must be carried out of the nucleus and into the cytoplasm. This process is called protein synthesis because we are synthesizing proteins. Protein synthesis takes place in two steps: transcription in the nucleus and translation in the cytoplasm. Transcription is the process of transcribing DNA to mRNA. We are still using nucleotides, just substitute U instead of using T's. In the cytoplasm, the nucleotide language has to be translated to amino acid language. In order to translate from one language to another, use the codon chart. The ribosome protein factories attach to mRNA strands. tRNAs transfer amino acids to produce a polypeptide, which will become a protein. Proteins can be denatured or unfolded by a change in temperature or pH. Since shape dictates function, if they are not folded correctly, they will not function. Enzymes are proteins. Enzymes speed up a chemical reaction in living organisms by lowering the energy necessary for the reaction to proceed. Enzymes do not get used up. They can be used over and over again. Mutations happen. If a substitution is made, the resulting protein may not even change, or it may function differently than the original. If an insertion or deletion is made, the resulting protein probably will not function at all. And remember, biology is a way of life.